The thing that I just really don't like is the panspermia idea, which is just displaces the question onto something else. So, Francis Crick, who co-discovered the structure of DNA, was a proponent of this theory, quite surprisingly. And I've always thought of him as being a very talented theoretician. He didn't really do that many experiments, actually, but he took it somewhat seriously, and I've never quite understood that.
Mike, Misha, and Xinghui talk origins of life with Jacob Fine, researcher in computational biology and molecular genetics at the University of Toronto. They discuss the necessary components of a good origin of life theory and how the field has evolved over the last 150 years. Please welcome Jacob Fine.
We decided to do something unconventional this time and film this in a nice Russian sauna. So we're not in the hot room. We just got out. We're kind of in the ante-room here, drinking tea. And, a little bit of inspiration was Darwin's original, he mentions the origin of life maybe happening in small warm pools, little warm ponds, in a letter to his friends. And so, we can take it from there. So Jacob, can you just give us, what are the core things that any solid origin of life theory needs to explain? What are the key things about life that any theory has to explain? Where did these things come from?
Great. So thanks, Mike, for having me on. Thanks, guys. It's great to be here. So one of the most essential parts of life is that any theory of the origin of life ought to consider is replication. Now, everything that exists, every living thing that exists, has emerged by descending from something else, and therefore is a sort of replicated version of that thing. And so everything on this, every living thing, if you just look at their parent, their parent, their parent, going all the way back, you can make a big tree, a tree of life, and you could trace that back to the first living thing, which we call the last universal common ancestor. And that thing had to have the property of self-replication. So it's a very important property in life. The other property of life that any theory ought to have in it is the property of compartmentalization. Now, all of life is, so, we ourselves are distinct from our environments. We have a system, and there's surroundings, right? So the system surroundings barrier has emerged a while ago during the origin of life. And, that is essential to modern life. So this idea that there's a compartment, and that compartment has its own energetic properties. So there's an entropic cost associated with forming a compartment, and that compartment is the basis for much of life, including the fact that all living things have cells, or maybe they're one cell, a unicellular organism. And all cells come from pre-existing cells, and so on and so forth. So that's the second property. The third property is the processing of energy. So, life it involves doing work to counteract the force of entropy in the second law of thermodynamics. So that's another essential property is the processing of energy from its environment. And the fourth thing, I mentioned replication. I'll talk about this more, is catalysis. It's essentially the process of speeding up chemical reactions to make them actually happen. Most chemical reactions, if they did not have a catalyst, would not happen because it would take too long. And so this speeds them up. So those four things, any theory of the origin of life ought to contain.
And when did people originally start seriously thinking about this in a scientific way? When did the origin of life become kind of a tractable, at least semi-tractable problem that people started thinking about?
So Darwin, being the pioneer of the theory of evolution that we understand today by natural selection, he was, of course, also interested in how did life itself start. We have life today. We can look at all the organisms, but a very natural question is, how did everything start? So Darwin, being a serious scientist, thought about this, but at his time, there still wasn't enough data to really model and make predictions about the origin of life. So Darwin thought about it. So one might say, it goes back, quite a long time, all the way to the days of Darwin in the late 1800s. But so for example, Darwin wrote to one of his colleagues after he published The Origin of Species, it is mere rubbish thinking at present of origin of life. Now, that was something that he said in his time, but quite a lot's changed since then. We've learned quite a lot about how the world works from a molecular perspective, from a computational perspective, and so much more has been learned. The next sort of major scientific thinking and work on the origin of life was done by two people living in very different places. One was JBS Haldane, who is a British population geneticist, and he thought about the origin of life. He did not, despite all of his mathematical rigor in his thinking of the origin of life, he didn't really develop a formal model of it. He sort of proposed the idea that small, subtle chemical reactions, if they happen over a prolonged period of time, can lead to life. And so that was what Haldane put forward. Another person in a very other different part of the world was Alexander Oparin, who was a Soviet biochemist. And he wrote a book in the 30s called The Origin of Life, where he proposed quite a similar theory as JBS Haldane. And this idea of the primordial soup really developed in his work. He was very chemistry focused, very metabolism focused. And so these two ideas, so Haldane and Oparin, their thinking became now known as what's now known as the Oparin-Haldane hypothesis, which is essentially what we all know in a sort of a pop science way as the primordial soup. Yes.
I was going to ask, since you mentioned there's like different approaches, different kind of views on the origin of life theories. So are there any contradicting ones? Like, let's say if you take cosmology, there is like a Big Bang theory and accelerated expansion of the universe, and then there is like a stationary one. Of course, now stationary one is put on the side. Is there anything in the field of origin of life where there's like several different approaches, competing ones?
Yes. So very much. So there's various theories. Some are contradictory, and I'm going to walk through those. So this actually leads perfectly into the next big steps, which leads to a theory which some have contested. So I'll walk towards that. So a key for the background of that, a key thing that happened in the 50s was the elucidation of the structure of DNA by Watson, Crick, Franklin, and Wilkins. And this really let us see that life, in fact, has what Schrodinger called aperiodic crystal that has a defined sequence which carries with it information. And this formalization of the physical basis of the gene, the unit of heredity, the thing that all life has genes. The formalization of this into this nice model really allowed us to think about sequence as being a key part of the origin of life. Now, this is where you get into the contesting theories. So with this theory of DNA being the sort of molecule of life, there were two other main informational molecules in life, which constitute what's called the central dogma of molecular biology. So one I said was DNA. The other two are RNA and protein. So these of course were known, but the relationships between them got really firmly understood in the 60s and 70s. And so given that life, all of life, whether you're a human or a tree or a bacterium, has this central dogma. You have DNA, which makes RNA, which makes protein. Now, the question is, since all of life has this, well, which came first? They didn't all pop into existence at once. They weren't suddenly intelligently designed in some short number of days, right?
It's like a chicken and egg problem.
Yes, exactly. So this gets to the chicken and egg problem, which leads to which theory has more plausibility. So, in the central dogma, which is the backbone of all of our understanding of molecular biology, is that DNA, which sits in the nucleus, makes RNA, and then the RNA leaves the nucleus and it goes to the cytoplasm, which is outside of the nucleus, and it's read by a machine called the ribosome, which then makes a protein chain, there is a mapping from nucleic acid sequence space, which is DNA and RNA, onto protein space, which is protein, and there's 20 letters of the protein alphabet, and there's 4 letters of the nucleic acid alphabet. And those 4 letters in the nucleic acid alphabet make up the 64 codons, some of which map onto the same amino acid. So different amino acids have different number of codons that specify them. So this is essentially sort of the backbone of molecular biology. And now, given all of that, the question is, which came first? Well, that leads to what we call the RNA world hypothesis, which is the theory that RNA came first. And there's a contending theory that would say that proteins came first. And these are two sort of competing theories. But what people have essentially, I would say the majority of molecular biologists now accept the RNA world hypothesis, which is a theory that RNA came first. And I can get into all the reasons for that. Yeah, so that's
that
essentially.
That would be great. Yeah, get into the
we'll get
reasons
into that,
why.
Yes.
Yeah.
So which religion do you belong to?
Yeah, I've heard the RNA world hypothesis be described as a religion, but it's based on quite a lot of evidence, so it's quite firmly established now.
What do you think is the top, like number one and number two? Number one and number two, strongest evidence. Great piece of strongest evidence.
So the sort of the nail in the definite the best piece of evidence is that the ribosome, which is the machine I mentioned that makes protein by reading an RNA template, it's a complicated machine, and most machines in cells are made of protein enzymes. They're complicated machines that they have what are called active sites, which essentially are the places where chemicals come to bond or ligands, and it breaks them apart or it puts them together. And most of them are made of protein. So naturally, people expected the ribosome's core function, its active site, to be made of protein. But they were quite surprised to find out that actually it was made of RNA. And so it's a catalytic RNA molecule that sits in the center of the ribosome. And the fact that protein synthesis is catalyzed by RNA almost necessarily implies that in the origins of protein synthesis and in the origins of the genetic code, the scaffold upon which it built on was made of RNA. It almost necessarily has to be true because you have to evolve the genetic code, to evolve life. The origins of life and the origins of the genetic code are tightly linked. You need this sort of RNA infrastructure to already have been in place. And when you look at extant life, all of life, whether you're a human or a tree or a worm or a bacterium, we have this RNA machine in every cell of our body. And if you actually look at the base pair sequence of it, it's almost 99.9% identical for this region. So we say that's universally conserved. And so if all of life has this, it necessarily follows that the beginning of life had this. All cars have wheels.
There's some kind of common ancestor that had the RNA machinery in the ribosome.
Correct.
When you present it like this, it seems very obvious that RNA should have come first. Yes. So what compels people to still hold on to the protein hypothesis that came first? Right. Like what are their arguments?
Yeah. So I'll address that, but I will first add also that the analogy, which to understand this, is like if all cars have wheels, then the first cars had wheels. And if all cars have steering wheels, then the first cars had steering wheels. But not all cars have electric windows. Some have roll-up windows. So, we don't know because that's not a universally conserved function. But in this case, it is a universally conserved. Yeah. So why protein? Why protein?
Well, you could imagine that their car started out with tracks and then people realized, we need wheels. But I think there
must have been a stage at which there was
a state. Well, in an evolutionary system, it's easier to believe that because there are no massive jumps, right? Like it has to kind of iterate towards something. But I think in a human design system, you could have big discontinuities, right? Yeah. But yeah, it makes sense that in an evolutionary system, if you see something that's 99.9, what's the typical conservation rate? Like if you have a different enzyme that's in all,
it ranges quite extensively, depending. Some genes are just human specific in the sense that their sequences are very divergent, and they could be 20% identical even with a mouse. But this is quite rare, and I will add that in the universal gene set of life, in this set of less than 100 genes that are found in all organisms, they're dominated by translation related. So translation being ribosome related things, given that indicating the ribosome is very old. It's probably the oldest thing. And so to answer the question about I guess I can move towards now talking about the protein first view. Okay. So one of the main arguments, well, first of all, our sample space is really three. We have DNA, RNA, and protein. There really aren't any other like main theories of what came first, because the genetic code the central dogma has three main information molecules. So if one is true, then the other has to be false.
And I guess just to double click on the word information, I don't know if you already mentioned this, but I think we were discussing this before, and it's an important point that like one of the fundamental functions of life is to carry information. Yeah. So even if you could imagine some kind of metabolic processes or something that came before information, they probably weren't like exactly alive in the sense that we care about, because we want to see information, something that evolution can act on, and information that passes along. So like that first informational system probably was either protein, DNA, or RNA, or some precursor that was like an aperiodic crystal of some kind.
Correct. And yeah, so and I would just add also that as for when life begins, it's quite a matter of definition. It's like asking sort of like, when did the first humans emerge? There was really a continuum of intermediates between the first humans and present humans. And of course, wherever one draws the line and says, oh, this is when it turned into a human, it's not really a binary classification problem. It's actually it's a quantitative trait, essentially. So life is a quantitative thing, and it's an informational thing. So, so now to get to the protein theory, it's important. So a lot of the arguments for the protein theory are actually arguments against the RNA theory. And so this is one of them. So, can RNA do enough functionally? In present life, we see most functions in the cell are, genes make RNA, DNA makes RNA, which makes proteins. And there's so many proteins, there's hundreds of thousands of proteins per cell, and they're really doing everything. And all the like the functions of catalyzing reactions, building things, making structures, that sort of thing. So one of the arguments against the RNA world was that, well, RNA just doesn't do all that stuff. RNA is more passive than protein. Protein's actually getting to do all these functions, and RNA, it doesn't have as big as what has been called a catalytic repertoire. It doesn't do as much. But that view is growing increasingly implausible, because we've learned in RNA biology, we've learned quite a lot about what RNA could do. It could do quite a lot, and it can do, in many cases, as much, if not more, than what protein can do. In many sort of theoretic ways of looking at life, is like RNA is like the center. So John Maddock, who's a great RNA biologist, wrote a book called RNA, the Epicenter of Genetic Information. So RNA is at the center of the information processing system of life, and it does quite a lot in terms of, it can act as a catalyst. And that's another key point. So Tom Cech and Sidney Altman in the early 80s published papers which showed that RNA can actually act as an enzyme, which people thought was limited to proteins, right? But, and that won the Nobel Prize. That led them to both win the Nobel Prize. So they showed that RNA can actually do this key thing we thought only proteins can do. And then the discovery that the ribosome's core is a ribozyme was after that. You asked me the two top reasons. The top reason is that the ribosome core is a ribozyme. The second best reason is that RNA can be a catalyst like protein.
It's the only single point which can do both.
Exactly. RNA can be both. Yes.
And the reason protein is a bad candidate is, from my understanding, is partially that it's hard to copy. Correct. It doesn't have this copying mechanism built in where you have bases that can stick together in two strands, whereas both RNA and DNA have that. Although DNA is better at it, but RNA, you can do it with RNA as well.
So there's another thing, like RNA. So we asked before, like, what are the necessary conditions of a theory of the origin of life? Well, we now established the genetic code, DNA, RNA, central dogma, DNA, RNA, and protein. And DNA is an informational molecule. It's purely informational in that it stores and transmits genetic information. And and proteins are the molecule of function. They perform the sort of the dirty work of the cell. But RNA is the epicenter. It's the middle of that. And now with what Czech and Altman showed, we know that RNA can it can do the job of protein by doing catalysis, and it can do the job of DNA as acting as an informational molecule. So it can do both. And not only that's just the proof of principle aspect, but the actual functional relic of the origin of life, you can look at it. You can go on the UCSC genome browser and look up the sequence of ribosomal RNA and look up and do the alignment and see that the alignment is like 99.9% the same across the tree of life. You could do it yourself. You could look it up and see it. It's there for everyone to see. So, you could also look at the studies that have been done on, like aligning, alignment. I should talk about multiple sequence alignment. So in bioinformatics, multiple sequence alignment is when you have a bunch of sequences from different organisms and you want to see how similar they are. So it uses a dynamic programming algorithm. There's a Smith-Waterman, there's a Needleman-Wunsch algorithm. And there's various ways essentially that it lines up the residues, and you can look at quite exactly how similar sequences are, and you can get like a percent identity from that. So it's like, so that's how we know. That's the actual like science behind how we know this.
I see. So speaking of maybe like replication abilities of RNA and in general like this thing in origins of life. So when you replicate something, inevitably you get errors. So let's say in like for our like computers, there's like Hamming codes.
Yes.
For quantum computers, there are like steam codes.
Yep.
So what preserves what does the error correction for?
And like the machine epsilon
for
like
RNAs and the living things.
So there's an error limit in biology, and it's called the Eigen's error threshold. So one of the key things about replication is it actually has to happen in a way such that the error rate does not exceed an upper bound, which Manfred Eigen, who was a great physical chemist, also won the Nobel Prize and worked a lot on the origin of life, a lot of theoretical work on the origin of life, worked out that essentially the length of the sequence is inversely proportional to the upper bound on the error rate. And the actual equation is that the error rate, let's call it R, is less than or equal to 1 over K, where K is the length of the first sequence. So that's the Eigen error threshold. And what happens if error rate exceeds that is you'll get what's called an error catastrophe. So the information in the population of replicating sequences rapidly gets lost, and what's called the master sequence, so the original template, is essentially abolished. So, it's important to employ concepts in statistics here because, when we think about sequences in sequence space, there's 4 letters in the alphabet of nucleic acids, A, T, C, and G. And so if you have a sequence of length K, then that means there's 4 times 4 times 4.
Do you want to do some derivations here?
I could be doing well, this is sort of simple. So I could do, I mean, the thing I was getting at, you want me to pull out?
We think.
Okay. All right. So let's consider a sequence of length K, which means it has K nucleic acids in it. 1, 2, 3, whatever, blah, blah, blah. Each position could be A, T, C, or G, right? And so there's 4 possibilities here. 4 here, 4 here, da, 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 da. And how many are there in total? 4 to the K, right? That's the number of possible sequences. And I really want to hammer home this point because it's so important. When we think about sequence space, we need to be really precise and talk about how big it is. So there's a total of 4 to the K possible sequences. That's quite a lot of sequences. If K is 5, I think that's what, 1,000. You want to get the calculator out? If K is 5, 4 to the 5 is 1,024, right?
4 to the 5?
Yeah. And then 4 to the 6 is 4,096, right?
Yeah, that's correct.
Great. Okay,
that's correct too.
Yeah. So
2 to the 10, yeah.
Yeah. And so like, okay, I really want to hammer home this point because we're talking about the origin of life as the origins of information. And what does that mean for there to be an origins of information? Well, So in information theory, there's something called the Shannon entropy, which is essentially the expected value of information in a sequence, in a sequence set of symbols formally. And DNA is actually a set of symbols because it's just the alphabet as size 4. There's A, T, C, and G, right? So the Shannon entropy is P, is you sum over the probability of each symbol, where Pi is so the ith symbol has a probability Pi. You just sum over all of them, and that gives you the Shannon entropy, and we call it H. So that's the Shannon entropy, and I want to narrow that down. And this quantity is maximized. This function has a maximum entropy if the distribution of symbols is uniform. So let's say the symbols, the random variable that denotes the sequence is denoted X. If it's a uniform distribution, meaning each symbol is equally probable, then the H is at a maximum. And so what that means is that if you have a uniform distribution so every sequence then has a probability of 1/4 to the K, that is maximum randomness. So this is our definition of what abiotic is. We now have a definition. It's that each symbol, each sequence in the 4 to the K space has an equal chance of being observed. And that's randomness. That's pure randomness. So what life is a deviation from that. It's essentially non-random, meaning deviation from uniform. And that was sort of the idea that my mentor Alan Moses and I worked on in our paper, where we proposed the RNA condensate model. And so life is essentially we're defining it as a deviation from a uniform distribution. And you could visualize this.
Yeah, how does it look like?
So yeah, if you just draw out a uniform distribution, each symbol, it's essentially just like a box, right? So that's a uniform distribution. This is the probability of X. That's X. So that's a uniform distribution. It has maximum entropy. That means each of the 4 to the K sequences is equally likely, right? Okay, well, now what is life? Well, life is when this distribution shifts to particular parts of that space. So let's draw it like this. So this is not exact, but I'm just giving you a rough idea that like when certain sequences are favored, you have a deviation, and then this gives you actually an exact number of bits of information added by a non-random, non-abiotic distribution of sequences. And this information has with it a cost, because no information can be created without energy. And energy and information are proportional, actually. And in our framework here,
and this comes from like the conversion of bits to entropy, basically.
Bits to energy. So for every number of bits, there's a certain number of joules. Actually, for every bit has with it Boltzmann's constant times temperature times the log of 2 number of bits, I think. Sorry, number of joules. That's right.
Per binary bit.
Yeah. And that's related to Boltzmann's, the KB times ln of the number of states. And a bit you could think of as a system with 2 states. So that's where you get this fundamental relationship. And it's the fundamentals of like, we're getting to like, what is life? Life is information. And we're defining what it is right here. So, okay. So that's that. Now, how do you get that? Well, we've actually formalized the problem now. We've defined what you need, right? Which is you need to have a gain of information over a random background. And yes, and so to get that, one thing you would need is to have replication of a master sequence. So this is from Manfred Eigen's framework, is that you have this like parent sequence from which everything descends from. And there's a certain error with every round of replication. Let's call the error rate R. And R cannot exceed 1 over K, where K is the length of the sequence. And turns out, so that if you use that, you now have a distribution of errors. And in computer science, when you have strings of equal length, the number of positions in which they differ is called the Hamming distance. And so it turns out the Hamming distance can be modeled as a random variable, and that follows a binomial distribution. So you could think of it as if there's K symbols, each one has a probability R of being incorrectly replicated. And so it's a binomial it follows a binomial because it's yes or no k times. So it's just k choose r times r to the k times 1 minus r to the power of k minus d. And that's the binomial for it. And if you use that and you combine that with, okay, another thing is if you have a sequence of length k, and you have a certain, if you have a sequence of length k, the number of possible sequences that are d symbols different from it has a specific form. It's k choose d times 3 to the d. And if you use that and you combine it with the binomial, you can now actually derive what p of x is. And you could actually quantify how different p of x has to be from the background distribution, which is the abiotic distribution. So you have an abiotic distribution, which is uniform, and then you have the non-abiotic, so the biotic distribution, which is non-uniform, and it follows this sort of like, it's not a binomial, but it's related to a binomial. It's a binomial multiplied by the Hamming distance.
One of the things when we were discussing this yesterday, one of the things that I found interesting is that you mentioned is that, and this is just like bringing it back to like empirical stuff, is that the human genome is roughly 3 billion base pairs, and the error rate of copying is roughly 10 to the 9th, so it's 1 in a billion. So actually that 1 over k number for the error rate threshold, the human genome is like three times lower than that threshold. So it's below the error rate threshold. That's theoretical. It's below. But interestingly, there are plants that are above, like a factor of 100 above the error rate threshold, roughly, but they might contain a lot of junk DNA that, like, when it gets copied, if there's an error, it doesn't actually affect the outcome as much. So that might be the explanation. But interestingly, there are things that actually break the break this rule.
Okay, so I'll elaborate on that. So yes, so it's true that plant genomes have error rates that might not fit clearly into this framework. But I'll explain. So sort of like two key things. So first key thing is that in the human genome, the error rate of DNA polymerase is about 1 in a billion. And the human genome is 3 billion bases approximately. And so therefore the error rate is, so again, the eigen gives you the max error rate. So we're still within the good limit, essentially. But when is it okay to pass the error rate? Well, it's okay to pass the error rate if not all the sequence is functional. So this is sort of like a key point. So we could think of the effective number of bases that are functional. So you could sort of like, think about it as if you're trying to like copy a book. When you copy a book, do you necessarily need the author's letter, or do you need the epilogue, or do you need the acknowledgement,
the cover,
the cover? Yeah. So like these are the less essential parts of it. And so Eigen's threshold really applies. So there can still be life as long as the functional stuff is okay.
So it seems like life is not perfect, and can we have life without errors, or errors is the essential part of?
Good question. Good question. So errors are an essential part of life. Without errors, there wouldn't be life, because you need errors to generate variance, genetic variance in particular, which is acted upon by selection. And selection picks among the variants for each quantitative or qualitative trait, which traits will increase their frequency over time. So you actually need to have mistakes, because the mistakes generate variety, and that's selected on.
But I think there's no lack of mistakes. Like, if you think of any origin of life theory, it's almost like you have to explain the low error rate rather than search for an error rate.
It actually reminds me, in like Gen AI, to generate something, you need like a noise distributions. I mean, usually they start like from Gauss noise, and then you can come up with some paintings, music, whatever.
Yeah. No, exactly. So like in a generative additive, GAN in ML.
Yeah.
So you have like two competing neural nets. One of them is just generating noise and the other is trying to critique. Yeah. So they have sort of like an arms race until they reach an equilibrium where they actually can generate an image, right? Or something.
Do we have GAN in Origins of Life?
Not to my knowledge, but I'm sure someone might find a clever way to use AI in the context of modeling the origin of life. Actually, so I'm actually doing some work on that right now. And we can talk about that later, but essentially, getting to this like information theoretic thing, like how much does the distribution of biotic polymers differ from the abiotic distribution? There's ways that you could model that, and you could sort of think of it as a classification problem, where you have like, how can you tell just by the sequence whether that polymer is in a biotic or abiotic class? So it's a binary class. You have abiotic or biotic. And so can you train a model to just read the sequence and essentially discriminate between the two? And can you do that with real data, which is, you might have like selects data, for example, like in vitro evolution, where they actually generate sequences. And can you train a model to tell apart the thing that was in the final step versus the first step?
Do you look into phase transitions from biotic to abiotic?
Right. So now that we've motivated the sort of the theoretical background of what it means for something to be life, we can now talk about how that might have happened. So I mentioned that, we have this like distribution P, living distribution, that differs from the background. Where does that energy come from? And it turns out one convenient way for that energy to be supplied is through a biomolecular condensate. And what that means is you have a set of sequences of polymers which, through their self interactions, separate themselves from their surroundings, and so the mixing of them in their surroundings is unfavorable thermodynamically. So they do what's called demixing. So they demix and they form blobs, and these blobs are called condensates. Now, and this gets back to Oparin. So Oparin, the Soviet biochemist who wrote the book Origin of Life, proposed that, well, he called them coacervates, were instrumental in the origin of life. And what a coacervate is, a condensate, I briefly described, but they're essentially, you could think of them as bubbles. They're bubbles that don't have a membrane. So in conventional biology, most things that are compartmentalized are bound by, they're encapsulated by lipids or phospholipids. They're encapsulated by molecules that generate a barrier between them and their environment, right? So what Oparin is saying is actually, no, you don't need that. You can have a simpler thing, which is just the bubble itself. It's that the small molecules themselves, through their own self interactions, can separate themselves and form droplets. And think about like when you pour oil into water, the oil droplets form. It's analogous. It's not the same chemistry, but it's similar. So that's essentially this idea of condensates. Now, this idea of condensates fell out of fashion for quite some time. Most biologists didn't pay much attention to it until roughly like 15 years ago, when molecular biologists found that actually condensates can function in cellular processes in current life. So it turns out that condensates in current biology can act to sequester things like enzymes and proteins, and that they can be dynamic in biological processes, and they can assist with like things like embryogenesis and development. And so they're actually quite important. So this was only appreciated recently with advances in like microscopy and sequencing and things of that sort.
So it was technological advance.
Yes, it's technological advance, and that led to a biological advance, which is understanding these condensates.
How big are they?
They're subcellular. So a cell is a couple of like, micrometers, and within that there's like so essentially subcellular. So quite small.
Tens of nanometers probably. So a single molecule is, let's say, 1 nanometer or something, like if it's small, or 3 nanometers if it's sizable. So it would be like, if you have a condensate of these molecules, maybe you end up with 10, 100 nanometers max.
So you would need like an electron microscope to look at it.
It probably depends a lot on the chemical conditions, like how big, like what's the stable size, right? Because this is another beautiful thing of it, is that if you grow one of these, eventually it breaks up.
Yes.
Because there's like a surface energy. Actually, it's a good question what the energy is that drives it to break up, but they end up splitting if they grow too much.
Yes. So I'll elaborate on a little bit of that more. So, there's these things called biomolecular condensates, which very recently have been appreciated in real biology. And so that's the background of so the origin of life. We have Operin talking about coacervates, but nobody paid much attention to that, unfortunately. And then we have, Watson and Crick solved, and Franklin and Wilkins solved the structure of DNA, which led us to think about sequence. And then we have people like Eigen, who gave us a theoretical formalization of the origin of life as that it's related to sequence. And then we have various experiments like by Saul Spiegelman, for example, who showed that you could replicate RNA in a test tube with a protein replicase. And so all these things are converging. And now, 15 years ago, you have, condensates being appreciated. So this leads to today. Approximately not today, but there's people have now started to think about in the past, I would say 3 years, that condensates might particularly be involved in the origin of life. Well, so I should say that is old, but what's been more appreciated is that biomolecular condensates are related to the RNA world, and that they could have been directly linked to the RNA world. And that is what
I think one point we kind of skimmed over. So there's two things. One thing is that we flipped the computation. It's actually the error rate by Eigen should be 1 out of 3 billion. And actually the error rate in the polymerase is 1 out of a billion. So it's still off by a little bit in the wrong direction. We kind of flipped it in our discussion.
We did,
I think we did that twice. We did it last night, and then the mistake carried through to this. But anyway, we just fixed it. But it's still roughly around there. But the other thing that I think we skimmed over, which is really important, is that any theory of an origin of life conventionally has to come up with a way to compartmentalize the reactions, because if the reactions are happening not in a compartment, they diffuse away. And so conventionally, you need a compartment and a system that's replicating itself. And somehow things have to diffuse through the compartment, and the right things have to diffuse in, the right things have to diffuse out.
Membranes.
The compartment has to split. So you end up with, the classic view is probably the Jack Szostak kind of approach, which is, you have RNA chemistry in a lipid membrane. But that in a way you have to stack. I think what's attractive, and when I read your paper and ended up contacting you, is what's attractive about the condensates is that it removes some of the, it has its own problems, but it removes this stacking of unlikely events where you have to stack up the RNA chemistry with the membrane, right? And these are maybe getting the right membrane that does the right stuff, plus getting, the chemistry, and having those things stacked seems like it's more difficult than having something that's inherently has its own segregation.
Yes.
Like the condensate gives you the membrane for free in a way, or something like
Correct.
a membrane.
So because compartmentalization is so important in life, system surrounding barrier, many people have spent a lot of time trying to essentially, so we have the replication problem, which we know has to be RNA because the RNA world. People have tried for a long time to get RNA to replicate in conjunction with a vesicle, which a vesicle is, could be fatty acids, essentially any sort of biomolecular thing that forms a bubble, and it actually has a membrane, it actually has a case, and we wanted to replicate that with an RNA inside, and we want the RNA to replicate with that. So we have two parallel processes here. It's very hard to couple them, and that's what great scientists like Jack Szostak and others have tried to work on and have succeeded in many ways. But there's still this challenge of trying to actually get template-directed RNA synthesis in conjunction with a vesicle replicating. And what the advantage of this condensate theory that Alan and I worked on and wrote about in our paper is that it solves both of these problems in the sense that, and then again, it's a theoretical paper. It's not yet experimentally confirmed, I should say. But what it really does is it says that, well, RNAs, could have separated themselves by their own self-self interactions. And that sort of allows for self-compartmentalization, independent of an additional process. So it simplifies things, it stacks things. And that's one aspect. The other aspect is this, and it's something we haven't yet, we talked about with the Eigen threshold, but there's a sort of another thing called the Eigen paradox, which is a result of the Eigen threshold, which is that, so we have the RNA world, right? But for there to be replicating RNA, that's still pretty complicated, right? And you need other replicating enzymes, we call them replicases, to replicate RNAs. And those, in an RNA world, they would also have to be made of RNA. but those, to our knowledge, replicases have to be still somewhat long. And again, length is sort of relative here, but roughly in the order of 100 to 150 nucleotides. And that's what a lot of the biochemists have worked on with trying to synthesize replicative RNA. They have to be pretty long. That's the short answer. And this length problem by eigen implies that the max error rate has to be roughly over 1 over 100 to 1 over 150. And that's a 1% or less error rate, which is actually quite hard to reach if you don't have an RNA replicase or any replicase. So that's the paradox. So how do we get that replication if we don't have the actual thing that we need to replicate it? So it's the chicken and egg. Now, this is where recent work on condensates has got really interesting. And this is what I was getting at within recent years. We've appreciated this. It's that condensates themselves can be catalytic. Boom, right? That is a revelation, that the condensate itself could be a catalyst. And this is what the serious polymer physicists and biophysicists have worked on, which is like showing that actually they form electric potentials that can do work, right? To actually make chemical reactions happen. And this has been shown very recently. Now, what Al and I did was draw a connection between this recent finding and the RNA world hypothesis to say that if you have an RNA-only condensate, then that condensate can be a catalyst itself. And if that's true, you don't need such a long enzyme. You don't need a 100 to 150 length ribozyme.
Mm-hmm.
The condensate itself can be a catalyst.
And that can be made of small sections.
And it can be made of small molecules that are much smaller than 100. So we know that. There's recent evidence that RNAs less than 20 nucleotides can form condensates, RNA only. And again, so we know that condensates can form made of just RNA, and we know that condensates can be catalytic, and we also now know, thanks to some recent work, that RNA only condensates can do simple catalysis. So it's quite interesting.
So just to understand better, so you are making hypothesis. Is it already on some observed data, or someone needs to go ahead and like make an experiment to prove your theory? I mean, maybe in general, like, would you, now you're on the computation side, would you consider, stepping out of your comfort zone and prove that your theory is correct?
So far, the most physical labor I've done in my PhD was assemble the chair on which I sit and code. But I'm not, my skill set is more in the computational and sort of like theoretical mathematical side of biology. We're open to collaborators, and we'd be excited to work on such a thing. And this very, very talented set of researchers.
Yeah, so I think what he glossed over is actually some of the evidence that he's talking about came out after their paper and was citing their paper. So basically, they inspired a group of biophysicists or polymer chemists, I guess, to actually produce RNA-only condensates and do catalysis with them.
I should say their work was done very well and independently of ours.
Yeah, so they might have already been working on this.
So Hoff and Dindo, those are the last names. They've done excellent work on essentially showing that RNA-only condensates can form with less than 20 nucleotides, and they can get simple catalysis to happen. They could speed up the reaction kinetics by a factor of 10.
But the $1 million question or thing that's missing is that they've done catalysis of certain reactions, but not of RNA synthesis. So in order for this to become like an evolutionary system, it has to catalyze RNA synthesis, and ideally not just random RNA synthesis, which you can still maybe live with, but you want to catalyze like copies of the thing that made the condensate, right? That would be ideal. And I think that's the big next, that's like the open thing that, if somebody could do would be an enormous breakthrough that's kind of driven by this theoretical, he's giving out Nobel Prizes. But that would be like the big breakthrough that can be driven by this. I mean, I guess there's another way that it can work, which is that you can have random generation of sequences, but then their selection, the condensate, that happens like somewhere else, right? And then the condensate just selects the ones that it likes.
Like mutations, maybe.
No, you just
Some
have random,
external
like
factors.
let's say, there's been a lot of chemistry work showing that, nucleotides can form in prebiotic conditions, kind of like
Yes.
old Earth conditions. And so basically the idea would be that let's say you're forming random sequences of 4 or 5 nucleotides or something, and then the condensate is choosing the ones that it likes. And so it grows just by absorbing them rather than copying them. So that's another way that it can work, but that's not been shown either. So, right? Like,
Yes.
correct me if I'm wrong. So showing some kind of mechanism where the condensate can grow.
Changes the
Polymerization.
statistics, right?
Yeah.
So you start from uniform distribution.
Yes, and then the
and then
condensate plays with
catalysis
the
would shape it in the right way.
exactly. And then basically, like the big question is, can somebody show if this theory is correct? And it does show like a path that's experimentally very interesting. Like in my opinion, when I read the paper, I was like, oh man, I wish I was doing experiments on this. Is can you do like some high throughput experimentation where you find a condition where you get one of these condensates that's able to either select or grow these short nucleotide sequences that basically copy it, with decent fidelity. But interestingly, the fidelity doesn't need to be that high because the error rate, this eigen threshold is basically, let's say if you have 5 nucleotide sequences, it's just 1 out of 5. So you need a 20% error rate,
So
which
you
is
need
still
very low
It's
error
much better
rates.
than random, right?
Yeah.
But it's actually very realistic, maybe.
Yeah.
Or at least it feels realistic compared to an enzyme. Like if an enzymatic process with error correction gives you 1 in a billion, like it feels, if you were asking for 1 in a million, that would be like a little bit scary, but 1 in 5 feels better.
Well, do we know what's the catalysis process? What it needs to be?
So getting to this mechanism that would be, groundbreaking if it was elucidated, definitely, which is that if you can show the $1 million question, if you can show that RNA undergoes replication in conjunction with a compartment, it could be a condensate according to our theory, but again, it could also be a vesicle if someone could show that it undergoes replication in conjunction with that, and that includes polymerization from a template. So when any nucleic acid is copied, it is copied in a way that, you have a template and each monomer is added one at a time in a 1-to-1 way on the template, right? And so this templated dependent process is essential for all life as we know it. If someone could show that happening in conjunction with the replication, growth, and division of a compartment, then that would be very groundbreaking. And so there is thermodynamics worked out of like what the energy cost of adding a nucleotide is onto a template, and it's like that stuff's known. And there's like various other sort of things known about like the stability of various base pairs and things of that sort. In the case of like secondary structure, there's something called the Turner rules, which is the rules of how pairs of bases that are adjacent with each other pair with other pairs of bases. And so this stuff is worked out. And what we need to now see is for this polymerization chemistry to be worked out, which is quite challenging. And that's one aspect. That's one possible mechanism. If this happened in conjunction with the condensate growth and division, upon which selection could be acted on, then the other aspect of it is that the condensate itself acts as a kind of classifier, a statistical classifier, where it can tell apart things that are self versus not self, because things that are self join the condensate and things that are not self do not join. And the more similar a sequence in the 4 to the k space is to itself, the more likely it is to join, and so on and so forth. So that is sort of this, polymerization independent selection. So it's sort of by selection, the condensate's able to maintain self identity independent of physical template based copying. And that would be interesting also if that was shown experimentally. And again, I'll add the caveat that the paper that Alan and I worked on was a theoretical biology paper. We developed a theoretical framework and we did some derivations, but we need experiments to really confirm this model. And that's now sort of where the next steps are.
And it seems like there were already experimentalists working on this. So to some degree, the timing is curious, which it often is in science, that a certain idea is ripe at that point. So do you think that these recent papers are going to give rise to an explosion in research in condensation? Are people excited about it? Surely Mike is excited about it and wants to spend his life now doing this.
Maybe it's Mike's
do you think
PhD
this is
topic.
Like, is this
finally.
excitation palpable?
It's very hard.
Yeah.
It's very hard to predict the impact of something. But what I will say is that, part of it, you're right, is the timing and like the trajectory. But also, I think, in the origin of life, the predominant origin of life community and the predominant bio community, the bio departments, there's sort of an attitude that's generally held by a lot of people, which is sort of like a reluctance to accept mathematical and theoretic methods, and sort of this tendency to, and not only that, just the theory in general is not emphasized as much in molecular biology. And this pattern sort of started in the 70s when they invented genetic cloning, I think. And before that, molecular biology was really created by physicists primarily in the 50s and 60s, who were very keen on making theoretic models and calculating things and deriving things. But really, once they got their claim to fame and all the success that happened in the 60s in molecular biology, we now have this period where genetic cloning was invented by Paul Berg and this took shape in the 70s, where biologists shifted their focus to just trying to clone everything and trying to really do all this physical labor. And there's nothing wrong with that, but it's just the theory sort of dwindled off. And this lack of theoretical emphasis continued to develop 70s, 80s. And most of the people that were trained in this period trained in a time where theory wasn't emphasized as strongly. And currently, most people running biology departments have this training. So there's essentially a lack of theoretical emphasis. And that's, I think, been a source of why there's been a lack of progress on the theoretical front. And I think now with the rise of computational biology, we're now at a time where we can really think about theory and actually try to go ahead and model. And of course, we need experiments just as much as we need theory. We need both. We really need both theory and experiment to really have the most impactful work that we can possibly have. Both are important. We need to do real biology, not just abstractions and sitting in an armchair. We need to do real work, and we need to make real.
So what constraint?
Yeah, but to this point, Manfred Eigen, like the guy from the Eigen threshold, error threshold, he actually has this famous quotation attributed to him, which is theory has the alternatives of being right and wrong. A model has a third possibility of being correct but irrelevant. So could you expand on what you think the point of a model, what makes a good model? What do you hope the function of a good model is going to perform?
Yes. So modeling is very important, but models shouldn't just be done for the sake of modeling and trying to be fancy with math. We should try to fit the data and make predictions that can be experimentally tested. Or alternatively, make predictions about existing data that allow us to understand something new about the data we already had.
Yeah, I think one interesting thing you do in the condensate paper is you basically try to figure out what is the number of nucleotides that you would expect in the condensate, given that I think this is an interesting concept you have, which is that the bigger the sequence, there's a happy medium, because the bigger the sequence, the more likely it is to form a condensate, and the more energy it releases when it forms a condensate. So you have more energy to do work. But as the sequence gets bigger, it's harder to copy. So there's like this trade-off that forms.
Can I have some water? Thank you. Thanks, Mike. So, we need to have, one of the theoretical predictions, real predictions that our model makes is on the length of the first genes. So that's a real thing that could be tested. Can genes of this length actually form sequences of this length actually form condensates? And so this thing I talked about, like here with the distributions, how deviant is a distribution from a uniform? The max deviance that a distribution can have from uniform is given by the energy of the condensate formation. So that actually sets an upper bound on the max deviation it can have. And if you compare those two distributions, you can make some plots and derive from that what the length of the polymers are as a function of the energy in the upper bound if you simulate it, right? So that's a prediction we make. And that's something that's important. And I want to just address the thing about like the development of biology. So so between the 70s and 90s, there was a lack of theoretical emphasis, but that all changed in the early 2000s when they sequenced the Human Genome Project. So now, and well, up until that point too, many people started to apply advanced math and theoretical modeling to understanding biological sequences. And that was instrumental in assembling the human genome. For example, to actually build a genome, you have to do it in a piecewise way. You would join smaller pieces until they join up and up and up. And there's an algorithmic way to do that. And so that's where computational biology really started to get its claim to fame again. And now biology departments do a lot of quantitative work and experiment and theoretical work. So that's also part of the motivation of like why we're here now is we've sort of biology's come back to appreciating quantitative rigor.
So what limits your computational models?
Experimental data. I think we need more. We need data with like condensates, RNA forming condensates, and understanding sequence space in real in vitro evolution. So the better data we have, the better models we can make.
Any like synthetic data approaches?
Yeah, we could do synthetic data and we can do that. But if we really want to understand like real biology, we need real biological data.
And so is this model truly prebiotic, or does it have to assume anything in terms of like fancy engineered RNA to work well?
Yes. So our model like makes certain assumptions. For example, we set the lengths of the polymers in our model are equal, but that doesn't matter that much in terms of the actual calculations. It's just easier to calculate distance because Hamming distance assumes equal length, and if you could generalize that to unequal length, but that's an example of like an assumption. But and also another assumption is the polymer physics. We just used like a simple model of polymer physics called the Flory-Huggins model, but that could easily be expanded on if there was to be an RNA theory of phase separation, which there currently isn't actually. Mm-hmm. So if there were to be such a theory, that would help like firm up what the upper bound is on the max energy, because we're using a theory that is I'm not a polymer physicist, I'll confess that. We had help from polymer physicists in like getting our paper read. So we, that's something that is very important, is firming up what this upper bound of energy is. So
and that means you mean the upper bound of the energy released by
the condensate forming, yeah.
And therefore the work it can do.
Correct, exactly. Because we can get so currently again,
polymer and the relationship. I think it's important to really clarify this is the work that the condensate can do in resulting from the energy it's released when it forms can be converted to information, because at least theoretically, there's like an energy cost to the production of information. Exactly. Therefore, you can basically make the claim at least that this work is available to produce information, right? To organize things non-randomly.
Exactly.
And that's like an interesting component, and that also relates to the error rate and everything else.
Exactly. So, firming up like what this delta G is that we were talking about. And I want to say, because so the other thing is all these communities of research are working very independently. So you have like biochemists who are grinding like 80-hour weeks, like in the lab, trying to just run experiments. And then you have theoretical polymer physicists who are running simulations and deriving equations and doing really cool things like that. And they're not really thinking about biology, even though they're working sometimes with biological data. And so they're not really interested in RNA necessarily. They don't really see why it's so important. And so that's why that's one reason why there hasn't been like an RNA theory of condensation. It shouldn't be that too hard for someone to go ahead and derive it. Actually, so we've got help from some very talented theoretical physicists that we've been talking to about this and are interested in like deriving these sort of things.
So this is the reason you're now here on the podcast, because then you can get all of this in front of potential collaborators and tell them this is an interesting problem to work on, and maybe not even a very hard one.
Yeah, and our paper's out there, so feel free to give it a read. Fine and Moses, 2025. and it's published in Journal of Molecular Biology.
What originally got you interested? So you seem like you would have been pretty happy doing pure math or something like that, or computer science, working on AI or something. What got you, kind of what hooked you to work on origins of life, or comp bio and origins of life specifically?
Yeah, thanks. That's a good question. So, in high school, my favorite two subjects were biology and math. And I was always interested in, so, we took like calculus and functions and all kinds of things like that. And, but I was always really, really curious about the natural world. I was always curious about science, and I wanted to be a scientist. That developed over time, but I was always definitely curious about science. And, they're really, there's, chemistry, physics, and biology are sort of the things. And I was like, okay, I guess I have to pick one of them. And so, I was very intrigued by the complexity of nature and the emergence of function and the emergence of form and the emergence of like, how the heck does an eye work? That really intrigued me. Those type of questions excited me. Like, how can an eye possibly evolve? It's crazy. That blew me away. I was more interested in that than like, how does light work, for example? Like, I just so happened to be gravitated to that. So I just so happened to pick that my major would be biology in undergrad. And then, towards the end, I was like, okay, well, I've always liked math. I took some math courses. I did some research that involved math. And I got to the end and I was like, okay, well, I really, I did an honors thesis that was like a bioinformatics project analyzing sequences of the origin of life to predict what the first genes looked like. And so that sort of motivated me to then want to do a PhD in computational biology. And the field of computational biology was not really as loud and dominant when I was in high school. So maybe I would have picked that as my major. I don't know. But I think many people sort of start in biology as an undergrad and then do their grad school in computational biology. So that's where I'm at now.
Tell us about how you spent a whole summer reading Origins of Life papers. I feel like that's if this theory turns out to be correct, that's like an amazing origin story of its own.
Yeah. So I spent a full summer doing nothing but reading about the origin of life and writing.
You got yourself funded to do this too,
yeah, I got funding,
which is impressive.
I got funding from the Canadian government to do research on the origin of life. And I just did nothing but read papers and write my first paper on the origin of life with my supervisor, Ron Perlman, at the time during my undergrad. And, that sort of like really formalized my interest in this. And yeah.
What's the best thing you read that summer? Like what was the best paper?
So
best
I could list you some authors actually. I think that, and I could tell you what their work was. So one thing that really, really resonated with me was work by I think he's a computational bio, whatever. He was a scientist, Anton Petrov, who did work on ribosomal RNA and modeling what the first. So essentially, making a model of. He really got me thinking about like ribosomal RNAs. You could look at all of them and figure out what like the origins were. And he did all this work on computational analyses of like sequences across the tree of life and genes across the tree of life, and this sort of like organism-wide, species-wide analyses, like the universal gene set of life, and thinking about the origin of life computationally. So that stuck with me later.
Is it an old paper or is
so this paper was from
ones?
This was from the so this paper was specifically on the origins of the ribosome, and it analyzed ribosomal RNAs across the tree of life, and that really resonated with me.
What year? When was?
I think it was like 2014 or 2015.
Oh, okay.
Some
years ago.
Yeah. So that was, and I was in 2018 at the time. So this was my
2018, okay.
my undergrad reading. So it was not so far off. Sorry, not 2018. What am I saying? It was 2022. Anyway, so putting that aside, so that inspired me. The other person was Eugene Koonin, who is also a renowned computational biologist, and he's done all this like high throughput, organism-wide, species-wide, genome-wide, like look at all the data we have and just like come up with something. What did the first polymerase look like? Well, let's look at all the polymerases and then figure out what the common thing is there. So that work really, so those two people resonated with me in my senior year of undergrad. And that led me to like pursue work now in like high throughput computational biology, analyzing all these data sets and trying to come up with like theories to explain all of this stuff that we see. So those two people, I would say.
What's the most like of the like lower probability origin of life theories? Do you have a favorite? Like there's the hot vents. There's like life coming from Mars instead of Earth, right? So life falling on Earth as a meteor.
That just shifts the problem.
Exactly. So again, a lot of these things are also like, you could think of the origin of life as a timeline. It's not a single event. So you start off with like prebiotic chemistry, the simplest, how do you get like a complicated small molecule from nothing? And that's like some of the really cool work that like Shostak and others are doing.
But nothing being like chemistry that you can imagine, like cyanides and things like that people have figured out probably existed at some point in like the warm pools or wherever on early Earth.
Right. And so to get to the point of like which theory is my favorite. So well, these are not all mutually exclusive. Like they could have come into play at different points in the timeline. But the thing that I just really don't like is the panspermia idea, which is because it really just begs the question. It shifts the burden of it just displaces the question onto something else. So, Francis Crick, who was like a famous biophysicist / molecular biologist who co-discovered the structure of DNA, was a proponent of this theory, quite surprisingly. And I've always thought of him as being a very talented theoretician. He didn't really do that many experiments actually. And he was originally trained as a physicist and he transitioned into biology. So he was very good. But he proposed panspermia. So or took it somewhat seriously. And I've never quite understood that.
Yeah.
Guys, we should sauna. And then do another
Let's do
a sauna and then we'll do
Let's do some
the next
origins
section.
of life and the plot.
Do we need to?
All right, we're back here from the we did another steam another rounds. How do you feel?
Feel good.
Okay, nice. So here's a kind of a question, a little bit different question, more of a meta question. There's this interview that I recently caught a snippet of an interview with Peter Thiel with the New York Times, which I thought was like a cool crossover of worlds that rarely meet. And he said this one thing that in this clip that was really striking, which is that as a society, we used to talk about moonshots like as something that we were doing, right? And now moonshots are used as a way of describing something that's never going to happen. Oh, that's a moonshot. And the same thing for the Apollo program. Like we used to talk about doing the Apollo program, and now people use it in a negative sense. They say, well, this isn't going to require, we can do this. It's not going to require an Apollo program. Or the opposite, like we can't do this. It would take a whole Apollo program or a Manhattan Project, right? Or whatever. So yeah, my question to you is that what's needed to figure out the origin of life? Like, do we need a Manhattan Project to find the origin of life? Like what's really needed?
I would say a current problem in the field of the origin of life is there's many different people working on it in different departments doing different things that are not overall coordinated into some joint effort. And I think that if efforts were combined more cohesively, then that would be a very positive force for actually having high impact origin of life research. So in that sense, yes, I do think there should be some sort of Apollo program, so to speak.
Is it a question of funding? Because we are spending hundreds of billions of dollars for quantum computing, for example, but this is like the most compelling question that you could imagine, like the origin of life or the origin of universe, origin of life. Yeah. These big questions, why don't we pour money into that?
Right. Great. So, there is a funding problem for sure. And it is very... is it expected or not? The origin of life is a very fundamental question, yet there is not a major research focus on it in terms of the funding.
It feels like a lot of origin of life research is done by scientists that have already succeeded, so they have a little bit of leeway to like work on something fun.
Correct.
Right. So it's kind of unique, actually. You're one of the.
I'm early in my
retirement project. You've done your major stuff.
Yeah.
You got your career done.
It's like it's something you get to work on if you win a Nobel Prize or something like that. Right?
Well, so I'm still a scientist in training. I'm still doing my PhD. So I've been working on this sort of as a side project essentially, and I've been doing it since I've been thinking about it since high school really, and I did some work on it in my undergrad, which was full time. But yeah, I think we would really benefit from there being a concerted effort with large scale sort of collaborative projects uniting people from different backgrounds. And, especially I think it shouldn't be dominated by any particular type of scientist. I think all scientists are interested in big questions. I mean, to some extent, interested in big questions at least. And the origin of life is one of the biggest mysteries of all. So many people should be interested, and many physicists have thought about the origin of life. Computer scientists like Alan Turing and John von Neumann have thought about the origin of life. Freeman Dyson has written an excellent book on the origin of life. And it should be a collaborative effort of scientists, not any particular like biochemists dominating a particular aspect of the origin of life. It's a collective collaborative effort. And I think that would be a very positive development. So I think, there should be more funding into this sort of basic area of research. It's one of the biggest questions that any of us could ask. But that being said, there still are many, many expert leading scientists who have spent their careers working on the origin of life, even though they hadn't won Nobel Prizes. And so there's been some very excellent work done by people like that. In particular, for example, people doing work on in vitro evolution. So so another practical way you can go about doing origin of life research is by tying it in with real biology that people, not that this is real too, but tying it in with like goals that many people would have. So here's an example.
Goals that coincide with pharma, basically.
Correct. So here's an example. So the mRNA vaccines were essentially lipid nanoparticles with an RNA inside. That's a condensate. That is a vesicle with RNA in it. I mean, it's obviously very different biochemically than what the origin of life would have been, and in particular because the bases in the RNA are modified chemically, some of them, to stabilize the molecule. But that is an example of something where there's a link. Another link is in generating functional RNAs out of a random space. So I talked about there being 4 to the K possible RNAs. A large part of biochemical, certain biotech research. So Manfred Eigen coined the term evolutionary biotechnology. So essentially using principles of evolutionary biology to work on biotech. So like given a bunch of RNAs, you can evolve them to do things that are useful, like act as aptamers, act as things to bind drug targets, essentially. So that is functional research. And some people doing origin of life research also do this sort of to like add to their goals. And so I think, because this is such a fundamental question, like there are going to be lots of, we see this time and time again is basic research produces functional results that are very useful. So, that's something to think about.
So let's say if you were given unlimited resources, what would you still continue doing your research, or would you start another moonshot?
So I'm I'm working on a problem that I'm very passionate about. So I would still continue that. I'm very passionate about decoding the human genome, which is my main sort of research. The DNA genome was sequenced in the early 2000s, and that was completed. So we have the sequence. Now that we have the sequence, the question is, what does it do? And that was part of what's called the ENCODE project, and that's still ongoing. So I'm very much interested in that. I think that's also a very big question, a very important question. But admittedly, I do spend less time than I'd like working on the origin of life because there isn't enough expertise around me, for example, that work, like nobody in my department or other departments are really working on it. And so, there are places of that sort of thing, like the Santa Fe Institute, for example, historically has had a very strong theoretical biology, like under Stuart Kauffman working on the origin of life. And that stuff's pretty rare though. Right. And so I think we need more stuff like that. Right. We need more sort of like interdisciplinary systems level complexity science type institutes. Like, for example, like the Santa Fe, for example.
Is there a roadmap for origin of life research? So, for instance, in astrophysics, there's the astrodecadal. It's a roadmap for what is important the next 10 years. Does something like this even exist in origin of life research, where someone has mapped out these are the questions? Yeah, we need to answer. These are the experiments we need to do, so that people. Because for a lot of people, as you said, they come from different fields, but if they have a template like that, they can pick and choose.
Yeah. So there isn't really such a thing like that everyone's aware of, like the 10 commandments of the origin of life, like what we have to do next. There isn't really that sort of thing. And, part of that is because lots of people are doing it in their free time, or they're doing it as sort of a side project, or they're already famous from something else. And this is sort of their like their retirement project essentially. So, there's a lot of different things like that, but yeah, we would, that would be very, very useful.
You should start assembling that. We should start assembling that. Mike's going to fund it.
Well,
we could consider that.
by the way, do you have any like thoughts how to make the field sexier?
In what sense?
What do you mean? It is sexy.
Well, you said that somehow you need more people to come in,
Right.
so it's like somehow research is something holds back.
Yeah, I think part of it is there's sort of a siloed in approach that many like experts take, that like, oh, you don't have the background in something, or you're not in our field, like biochemists saying like, oh, you're not a biochemist. So for example, Chargaff, who's a famous biochemist, who developed the Chargaff base pairing rules, which, that A pairs with T and C pairs with G. He famously like dismissed Watson and Crick for being amateurs. Like, oh, you guys aren't formally trained biochemists, so get out of here. Like, why are you wasting my time? You don't even know the difference between a purine and a pyrimidine. Like, why are you wasting my time? And that approach we need to totally eliminate, I think, from science. It's very distracting and it holds people back. And that's why interdisciplinary science is very important. And I think everyone from different backgrounds should work together to solve these cutting edge problems. That being said, knowing what you're doing is very important. Like, training is important, of course, but we need to sort of be more loose in how we define fields and the boundaries between fields. That would attract more people in, essentially. So I think that would be an example of how to make it more appealing, let's say.
I think also like I have some thoughts on this. I've been thinking about Origin of Life a lot, and like searching for even like for guests for the podcast and so on about origins.
How do we increase the number of viewers?
Yeah. How do we get the
Origin of Life episodes?
How do we get the Origin of Life episodes to have the same number of viewers as the quantum computing episode? I mean, I think part of it is also that people don't feel. I think this is the kind of problem that has this feeling that it's always 30 years away, right? And I do think that like the solution is always 30 years away. So I think also just getting this kind of thing like a roadmap and just more understanding of like what are the things that have to be done to really crack the case, and do we even know them? Because sometimes if you don't even know what to do, like we still have to discover what needs to be done. Right. That's naturally going to be less. Like I think in quantum computing, everybody knows like we need lower error rates, we need more qubits. Like it's really clear, and those kinds of things feel to people like, okay, I know what I need to do.
And
Let's
the
get
big players are involved.
Yeah, that's true, and it also is connected. There's what it really is like. There needs to be like a Shor's algorithm kind of thing where everybody knows like if this is cracked, something very valuable comes out of it, and I think answering the big questions is very valuable, but it's kind of vague. So maybe we also need to think about like, if we could synthesize life, right, from abiotic sources, what does that give us, right? And there's a lot of interesting, like you can speculate a lot on that. Yeah.
Well, like first of all, like this sort of relates to a general thing of just basic science in general, because many people are like, oh, well, what's it going to give us? What's it going to apply? But one of the most fundamental things in basic science is you don't know what it's going to give you. You sort of need like deep research to be done in it. You need these large R&D projects that you don't know what the result is going to be in 5 years. Maybe
sometimes science fiction plays this role. Like certain science fiction books come true because people read them. Like there's not enough science.
For example, AI.
Yeah, there's like 100 examples of this, right? Right. So like even rockets and many things came out of science fiction.
Right.
And so I even think like maybe there's a role for science fiction to play.
Yeah.
Or you can maybe. I was thinking about like bringing bigger players, maybe like NVIDIA, if their GPUs can help.
Run simulations. Run simulations.
You need more serious simulations.
Yeah, for sure. That's very important. I think simulations, especially about the condensate model, would be helpful because we could like run molecular evolution simulations, and also simulate it under different conditions, like different distributions of length and free energy and sort of error rates and all kinds of things like that. We could try different things and also we could do things like calculate the probability of the origin of life, for example, which, and that's another question about like the nature of probability. But anyway, so that's I think would be a great idea, is getting sort of big players behind it.
Do you think growing up in like we were chatting before this, and you grew up in a religious Jewish background, right? And do you think growing up in that environment, obviously you're not in it anymore, but I'm sure that affected you a lot. And so like, do you think growing up in that environment caused you to be more interested in Genesis-like questions, like these questions of like origin or fundamental kind of like what are we doing here type questions?
So yeah, so I grew up in a modern Orthodox Jewish community, and I was taught things like Moses parted the waters. I was taught things like Moses parted the waters, and people live to be 700 years old and that kind of thing. And that was presented as sort of being like a scientific hypothesis about the origins of humanity. And I was, of course, very interested in that sort of thing. And I think maybe growing up in this environment made me interested in like those sort of questions, like how did we get here? Where did we come from? Who are we? Where are we going? Is there a purpose? And that was routinely part of my education and my upbringing. So I definitely thought about that a lot. However, the hypotheses I was presented by educators were that of a sort that didn't really rest on many firm foundations. So, I was always passionate about science since I was like very, very young, like 8, 7 years old. Um, you know, that I've always been interested in that. It was really only like towards the end of high school where I began to see a clear contradiction between the theory of how we got here according to evolution and science and the theory proposed essentially by the ancient Israelites, which was developed before the scientific revolution. So of course that theory is not going to be as accurate. And so, that's sort of where I am now, is being instilled with this appreciation for uh, the big questions, but now coming at it with a scientific perspective. So so I would say in a nutshell, it gave me an appreciation for the big questions, but not the tools originally. And I got the tools through education. And that's why I think education and science communication are so important, especially like people who are inspiring out there, like one of my original inspirations, Carl Sagan. So watching his show Cosmos and reading a couple of his books, and also reading various books on evolution and all kinds of things like that, that really inspired me. So I think that's very important, and sort of like having that message out there is very important. And but in terms of like the practical aspects, like you know we the religion I happen to grow up in does support like debate and intellectual inquiry and that sort of thing. So that was encouraged, and debate was very encouraged and promoted, and even asking questions about fundamental things like did the sea really split? So that was there. And so that's essentially it. So I think overall I'm happy with it. It's just that now I've sort of developed a different set of tools. Yeah, and that's essentially it.
So you also mentioned earlier that when you were like in high school or even earlier, you were fascinated by nature, by the laws of physics, by maybe how the world works.
Yeah.
So do you believe that this mostly comes from like noise, statistics, permutations, or there is maybe like some seeds of like someone created it?
Yeah, very good question. Yeah. Essentially, you're asking, is it random or determined in some sense? Is there some pre-programming to the universe? Yeah. Okay, so first let me just say that the answer to that question has no bearing on whether or not I have to keep meat and milk separate, let's say, by the laws of kosher. So that's a different question. So now that we move past that, we can ask like, is there any evidence that there's some sort of like design in the universe? And now that really, I would say, depends on what one means by design. So if what one means by design is that there's sort of these elegant mathematical structures that emerge, like the golden ratio, like Euler's constant, pi, and the relationship of pi and Euler's constant, right? So that sort of thing, like that elegant mathematical structure to nature, if that means design, then then you can call it design, and that's okay. I think that exists. And so there's sort of some necessity there. There's some sort of mathematical necessity. I believe in that sort of thing. That's what Spinoza believed. That's what Einstein believed. And so I don't think that needs to be called anything special. I think that's just what it is. It's this elegant thing that exists in nature. And there also might be some sort of thing of like emergent properties that are sort of built in. So by that, I mean as follows. So the outcome of any particular event at a given point of time could be random. So like flipping a coin, let's say, or maybe not, maybe Laplace's demon would say otherwise. But essentially, let's just say that Well, there actually are sort of like probabilistic facts that guarantee some sort of emergence. And here's an example of one. So if you flip a coin once, you don't know. But if you flip a coin, a million times, you could be sure that there will be an emergent thing, which is about half of them will be heads, assuming it's a fair coin. And so that sort of thing, these emergent probabilistic properties of nature, I think indicate some sort of order that's in the structure of nature. One thing that I'm always fascinated by is like how statistics explain nature so well. We see things forming specific distributions, just so to speak. Like if you go to the gym, like I was working on the gym and I paid attention to the distribution of like when you put the rod back in the machine to select the weight you want to do when you pull it down, you could actually see it forms a normal distribution.
The scratches,
and that's
right?
The scratches, yeah. The scratches form they're centered around a mean. And like,
and it has a simple mathematical formula.
Yeah, it has a simple mathematical formula. So these things exist. They're all over the place. And so that exists. That's there, sure. But does that mean I have to, again, separate meat and milk? No.
Cool. Yeah. All right. I think we can wrap it up on that, right?
Sounds great.
Thank you so much for having me.
Yeah. This was fun.
Yeah. Thanks, guys.
Amazing conversation.
Great stuff. All right. Okay.